This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 44 for August 7th, 2009. Hi, everyone. It's Vincent Racaniello. It's a beautiful sunny day here in New York City, and I've got a great crew to talk about this week's stories and viruses. And joining me here in New York City is Dick Despommier.
Hey, Vince.
How are you, Dick?
Doing very well.
Also in my office is a first-time visitor to TWiV, Jennifer Drahos. Welcome, Jen.
Thanks. Thanks for having me on your show.
Jen is a PhD student in my lab, about to leave, defended a few months ago, on to bigger and better things, and we'll talk to her all about that. But let's bring on, from the forests of western Massachusetts, Alan Dove.
Yes, it's a lovely day here, too. I was just whacking some weeds this morning.
Hey.
Interesting. See any interesting viruses in those weeds?
Lots of mosquitoes. I didn't really survey them, but they could have been carrying some.
Well, the Eastern equine encephalitis season is approaching, so watch your wazoo.
Well, I was in the Adirondacks 2 weeks ago, and there were tons of viruses, and all I could think about were...
You mean mosquitoes?
Mosquitoes. All I could think about were viruses.
Is it black fly season up there yet?
No,
passed.
it's passed, fortunately, but it is mosquito season. There's lots of standing water up there.
Yeah.
There's just marshes everywhere, and it rains several
very
days.
rainy.
It's just... And some of the kids are just covered with bites.
But notice, Vince, there hasn't been much in the way of West Nile virus yet.
Not yet.
Wonder why.
Why is that, Dick?
Well,
That be because it's such a wet year?
it's been wet and cold.
Yeah.
And the mosquitoes are as happy as clams with all them boids hanging around.
Well, now it's getting hot again, right?
A little
Or
bit.
it's getting hot. Finally,
warming
it's
up.
August. Here, the summer is almost finished, and it might get warm. But anyway,
We can only hope.
we have a bunch of stories today. But first, I wanted to just talk a little with Jen, because we have lots of listeners who like to hear about alternative ways to get into virology. So I wanted to ask you, now that you're finishing up here, how did you get here? What brought you? Now, you're from Iowa originally. What brought you to do a PhD in virology in New York City?
Good question. How I ended up in New York, I guess, starts from how I ended up in science. As a kid, I was always curious, of course, and it really all happened because of what my fellow friends and I called nerd camp. When I was about 12 or 13, I earned a scholarship to go to a summer camp called SciTag at ISU. No longer exists, but I took a genetics class. I took a genetics class when I was 13, before I even knew what a cell was. And for 6 hours a day, for 3 weeks, I was doing experiments and making E. coli grow in the dark. And I had this instructor, Jay Staker, who really taught me all I know. And I will never forget how to spell DNA because of him, which is "and" backwards.
Okay.
I was pretty much hooked after that. And this,
Never thought about it.
Yeah, it just turned into, in college, I worked my way through. I had a job in many labs, and had some amazing research experiences then. Realized then that the more I knew I didn't know anything. So I came on to graduate school here in New York City, which was a great place to be, both academically and personally.
We should point out that you started here in an HIV lab.
I did. I started in Dr. Jeremy Lubin's lab, studying mutations in capsid of HIV.
And of course, Jeremy's been on TWiV, and perhaps we'll hear you. And then when he left to go to Switzerland, you decided that wasn't in the cards for you. So I benefited by your joining my lab. Well, we have an AIDS story today, so that's appropriate. And then at the end of this session, we'll talk about where you're going.
Okay.
Because that's interesting, too, which shows that there's more than one career option, as we already know from Alan.
And a multitude of other virologists.
Especially from my lab. I've had quite a diverse series of different career paths, which was not an option when I was a student. There were one or two options at the most. You could go into academics or industry, but now there's much more.
Right.
So let us get on to our stories of the week. We have quite a bit. And the first is a very interesting story, isolation of genetically diverse Marburg viruses from Egyptian fruit bats.
Exactly.
Okay, so as everyone knows, Marburg is is a filovirus. It's a hemorrhagic fever-causing virus along with the Ebola viruses. And first emerged where, Dick?
I guess Marburg, Germany.
Marburg, Germany. And also,
it's the home of Herbst,
also
by the way.
Frankfurt and Belgrade in 1967. And some lab workers got
That's right.
infection from African green monkeys that had been brought in for lab research.
Yep.
And we never really knew what the reservoir of the virus was, although there were hints over the years because all of the sources seem to involve bats of some sort. So, for example, those monkeys that went to Marburg came from a place in Uganda where there were fruit bats. And there's other evidence as well. And so basically a group, this paper is published in PLOS Pathogens, and it involved the CDC, the World Health Organization, and various laboratories and ministries of health in Uganda. And the lead author is Jonathan Towner, who I knew because he was a student with Bert Semler, who is a coronavirologist at UC Irvine. And he went to the CDC after being a student there and is involved in discovering the reservoir of Marburg viruses. So this particular paper started in 2007 when a couple of miners in a Ugandan cave got hemorrhagic fever. And it turned out to be Marburg infection.
I didn't think they were supposed to employ minors.
It's that kind of show, isn't it? Or it must be just the time of the year.
There are no unions there, Alan.
Oh,
it's
that's
okay. You can have young people working.
That's right.
And it was these bats are infested. This is called the Kitaka Cave, and it's infested with Egyptian fruit bats. So the CDC and Ugandan scientists went there and they trapped bats. And this cave has a population of 100,000 bats. And they figure that about 5,000 of them are positive for Marburg virus. So in the end, they amplified viral RNA from tissues. And they also isolated virus from a couple of bats in the course of a year or so. So this shows that the reservoir is the fruit bat. And it's probably how those miners got infection.
Yep.
So the reservoir of Marburg and probably Ebola will turn out to be a bat, although that hasn't been established yet. But I would guess it's the same thing. And now this explains many of the outbreaks of Marburg infection. So it seems to me that there are apparently a bunch of these caves in Uganda. And Dick, would you know throughout Africa, are there such bat-infested caves?
There's tons of fruit bats all over Africa, yeah. Peter Daszak, who works for the World Wildlife Fund, has given us lectures in our emerging infections course with Steve Marston and I run. And his claim, and I think I've mentioned this before on the air, that there are about 20% of all the mammals are bats. And they're a very unusual group. And all the nasty viruses, as far as I can tell, are from bats, except maybe HIV. But, I mean, these are really fulminating, rapidly disseminating, disease-producing viruses.
I had a question. What is the mode of transmission for
aerosol?
It's aerosol. Oh.
Yeah, but rabies from vampire bats is you have to actually be bitten. But you can get aerosolized rabies also if the cave is actually heavily infested with bats that carry this. You can catch it by inhalation.
In fact, there was a story this week I didn't include because we have too much. 2 children in the western U.S. got rabies from a bat that actually bit them.
Yep.
So the bats were out during the day,
that's right,
which is a sign that they're
unusual. Crazy. That's right.
And they bit the kids, and they got the bats and turned out to be positive for rabies. So it's aerosol. So you really shouldn't be spelunking, right?
Well, there's tons of spelunkers, and not a lot of them get sick. So I'm assuming that it's not as easy to get as we might imagine. Just the same. By the way, there's another disease that I'm totally concerned about here, and that's E. Coli O157, which is a bacteria, of course. But just think of all the people working in slaughterhouses. And how come they don't get sick from that? Because they must be in contact with it all the time. So it's possible that if you looked in their serum, you might find very high antibody levels today.
Could be, sure. But
so maybe spelunkers.
do you think that American fruit bats, are there fruit bats in the U.S.? No,
we don't have any.
But where else in the world besides Africa?
Well, throughout South and Central America, there's a lot of fruit bats.
Australia.
Oh,
Southeast Asia.
Definitely. There's tons of them throughout the South Pacific.
Yeah, I think they're the origin of Hendra and Nipah.
They're called
viruses.
flying foxes. They're so big. I've
Right.
actually seen them. They're quite amazing animals.
So do you think that's why we don't have Marburg in the U.S.? We don't have the right bat species? Or could it adapt to a bat that's here?
Well, you could say that, but then Marburg isn't found anywhere. It's found there. And
So why doesn't it spread? That's the question.
Well, I think because bats are pretty local, all right? They're locavores. They eat local fruits, they eat local insects, they stay in groups that don't necessarily cross international borders and go all over the place.
So if
most people don't,
and they don't migrate. Bats don't migrate, by the way.
Right, and most people don't have extensive contact with bats.
No, although there are people that eat them.
Sure.
So if a person came to the U.S. with Marburg, it would stay.
Ozzy Osbourne, for example.
Right, exactly.
But it would stay in that person and not be transmitted to the bat population.
I think that's right.
So it's unlikely that this will spread to the U.S.
Very unlikely.
At least to the bat population.
That's right. And we get sick so fast that's the reason why these things don't get loose, like HIV, for instance. It takes years to incubate.
So I wonder if there are other bats, there are other viruses that are carried in American bats that we don't know about.
There must be tons.
And it would be worth looking.
Well, there is a bat survey going on right now. It's funny you should ask, but there is. Because of these numbers of, SARS, rabies, Ebola now.
Who's doing that? The CDC? USDA?
I think it is with the CDC, but this World Wildlife Federation is also interested in that. Because they want to protect bats, obviously.
Right.
So it's going to be harder and harder to protect a bat that is going to give you Ebola.
Well, that's another consequence of this report, which has been picked up by the press, is that they're going to like bats even less.
That's right. And they're great for controlling insects and that sort of thing. So it's a shame they're getting a very bad press all the way around.
Interesting anyway. So that's a paper in PLOS. We'll put a link to that. And that's the reservoir of Marburg.
By the way, there's another larger lesson to take home from this, and that is that if bats are solitary in terms of their colonies and they don't often mix with people, it means that by their own ecological behavior, they have isolated themselves apart from the rest of the animal world. They feed at night. They probably only eat rotten fruit that falls to the ground, basically, which is true. So that you could liken them to an island population of humans that have evolved their own specialized immune system, their own specialized diseases, and then all of a sudden, either they encounter an organism with an organism that could wipe them out, or they have an organism that could wipe you out, because it's been isolated from us for so long culturally. I think what you're seeing now with bats is more and more encroachment into their natural world, and the more that happens, the more these new diseases will emerge.
So, for example, the miners here,
they're
going after gold, I think.
Exactly.
One thing that reminds me that they mentioned in this article that these bats can fly about 300 miles.
Yep.
And they probably interact with other bat colonies throughout Africa. So this virus could be in all bat colonies.
It could.
Right?
It could.
And this colony of 100,000, they estimate they could have the number of offspring a year they could have is phenomenal.
Right.
I forgot the number, but
fruit bats obviously eat fruit, but fruit in the tropics that doesn't grow like a crop. So every fig tree, by the way, that's spread throughout, let's say, a tropical forest, will bloom and fruit at a different time. It's not synchronized. So you'll have one tree, another tree, another tree, and in different places, and the bats and the monkeys and the other birds can smell the ripening fruit, and they home in on that particular tree. So it's not like like a cultured crop of, let's say, apricots or something like that.
Got it.
However, when we do encroach and start mango plantations, that's when we do attract a lot of fruit bats into one place, and that is synchronized. And that's indeed how the Nipah virus got started.
Right. Okay.
Enough.
All right, next story.
Time for me to go home.
No, it's never enough. We can keep going. It's just...
no, but, sometimes I get verbose, and I probably should shut up.
No one's complained.
Talk to my wife.
I don't think she listens to the podcast.
She doesn't have to. She said, "Dear, what happened today?" We had a wonderful podcast. Let me tell you all about it.
right, now the next story I thought was about a plant virus, but it turns out not to be. So it will still be worth discussing. The headline in a paper, in an article that I found, is "Inspectors Find Potentially Devastating Citrus Virus in Psyllid Found at Fresno Shipping Site." All right, so basically a FedEx shipping facility in Fresno, California, they have dogs to sniff the packages, I guess for drugs and also plants because you don't want to bring in plants. And they found a package that turned out to have clove leaves in it. And in this article, they say the clove leaves contained this insect, which is kind of like a louse of insects. It's called a Psyllid. It's a sap-sucking insect.
Like an aphid, perhaps.
Sort of, yeah, I guess. And they convey this disease of citrus, which is called Huanglongbing, okay, Huanglongbing, or citrus greening. It's killed tens of thousands of acres of citrus across Florida and Brazil. Huanglongbing is apparently a bacteria-caused disease, which is transmitted by a psyllid. This kind of
so that's the vector
louse.
for the bacteria. Yeah.
And they call it a virus in this article, but I'm not sure if they're so citrus tristeza virus also causes a devastating disease of citrus crops. I don't know if they're talking about this here because the particular vector they're talking about is not one that carries that virus. It carries the bacterium.
Right.
And the bacterium, by the way, is Candidatus Liberibacter asiaticus, which is carried by a psyllid.
And there's also an Africanus species found in Africa.
So I don't know if they're referring to the virus or the bacterium, and they're confused. What do you think about this, Alan? Any ideas?
They are definitely talking about the bacterium. So I did a few quick Googles on this. It is the bacterium that causes this disease that's the big concern, and that has gotten into Florida. It's endemic to Asia and parts of Africa and India at this point. And so there's a lot of concern. One of the reasons that you get asked when you go to many places, the USDA will ask, or the local agricultural officials will ask, "Are you carrying any plant material?"
Right. And the answer is always no.
The answer should always be no.
No matter what.
Yes.
Because otherwise you'll be there for 3 hours. They'll be strip searching you next.
Right. And no, the reason places like California are paranoid about this is that plants from other places that haven't been properly inspected can carry lots of diseases that could, for example, wipe out the citrus industry.
Right. Like the Mediterranean fruit fly.
Yes, like the Mediterranean fruit fly. Hawaii is probably the most paranoid place that I've been to for this.
It's too late.
Oh, it's too late, but they're
too
still,
late.
they're still fighting the good fight.
Yeah.
So Florida has had a big problem with this. They've had a lot of their citrus dying from this disease, and it's the vector for it has already been imported. Or an equivalent vector is available here anyway.
Right.
And this is it's a bug. It's in the hemipteran order, and it sucks on the plants. And in the process, it transfers this virus, which then spreads through the plant, and it essentially kills it off. So yeah, that was the big concern. And this package had apparently gone through one inspection facility, and then it was being checked again. And this dog sniffed. I don't know if it was sniffing the leaves or if it was sniffing the bugs, but it ended up finding the bug, which tested positive for Huanglongbing.
All right, so this is a bacterial infection in this case, and
it is.
The news story is just misnamed it. They call it a virus, but it's not.
One of those common press errors that sets me off.
Right. Yeah, I thought it was a nice. Well, anyway, citrus tristeza virus is a virus carried by an aphid, as you said. And that's a positive strand RNA virus, icosahedral. So a little bit of plant virology. And we'll try and do as many of these as we can because they're in the news. But this one is not a plant virus. It's a bacterium.
Yes.
Okay. All right. We have a couple more stories. But before we go on, a few words from our sponsors, Citrix, the makers of Go to My PC. And if you feel that you need to sit in traffic every day just to get to your office PC, do not do it anymore, because with Go to My PC, you can work from home from Madrid or Cairo or wherever you may be in the world.
Or Uganda.
Or not from a cave, because there won't be internet access there.
No.
You have to have internet access. So Go to My PC is a form of remote access that allows you to access all the information on your office computer from anywhere in the world with an internet connection. And it's incredibly secure and very simple. Even Dick Despommier could do it.
Really?
Dick, you can go to gotomypc.com/podcast.
Huh.
You fill out a few bits of information, click install, and it's automatically installed on your office computer. Then you're going to get a little code with that install, that number you remember, and then from any computer in the world, you open up a web browser, you go to gotomypc.com, you insert your code, and bingo, there is your desktop. You can access all your files, all your email, your network resources, assuming, of course, you've left your computer on. And knowing you probably turned it off.
And you've remembered that little number.
The number. That is another number to remember.
That's right.
Anyway, you can try all of this for free for 30 days. Just go to gotomypc.com/podcast. That's gotomypc.com/podcast. Free 30-day trial. We thank them for their support. Parvo viruses are back. Well, they probably never went anywhere.
Right. They never left. That's right.
In Michigan, a story, Pontiac and Inkster, 300 dogs have been sickened and died as a result of this virus infection that causes vomiting and bloody diarrhea. Can kill a puppy in a couple of days. And I think we discussed canine parvovirus on another TWiV, but this just popped up. I thought it would be worth reinforcing that one of the tools of teaching is repetition. It never hurts to repeat.
That's true.
And I find the same thing for myself. Parvoviruses are DNA viruses with icosahedral capsids and single-stranded DNA genomes. And there's an excellent vaccine against these viruses. And in this particular article, which is from the Michigan Messenger, they say that with this economy, many people can't afford the vaccine, which is between $10 and $80 per animal. Although they also go on to say that the disease treatment can be almost $3,000. So it seems to me that the vaccine is a good deal. This is a really interesting virus because the canine parvovirus only appeared in the 70s, I believe. And it was originally, or thought to be, a feline virus that jumped host species. And a few amino acid changes was all it took that allowed the virus to go from cats to dogs. And Colin Parrish at Cornell has studied this extensively. And these viruses are transmitted among animals by contact. And, of course, you know, dogs love to sniff feces, and that's one way that they can transmit it to each other or by contact with each other. So if you have a puppy...
What about vertical transmission, Vince?
I don't believe it's vertically transmitted.
Can you, if you immunize the female dog while she's pregnant, will that protect the offspring?
You know, for a period when the antibodies are transferred to the offspring, but they go away in a month or so, and then they're susceptible. So you have to, if you buy a puppy or get a puppy, you should have it immunized.
Absolutely. You gotta do it.
I could be wrong on this, but I think Oprah, her puppy died of parvovirus.
Absolutely.
Is that right?
Yep. Not too long ago, right?
Oh, dear.
She could have afforded the vaccine.
I was just going to say.
Actually, she had them vaccinated, and they
late?
think that it was too late, that the puppy had already been exposed.
Dear.
How much time do you need between a vaccine and the time you're protected?
With rabies, if the virus hasn't advanced past your waist
about 2
in your
weeks,
nervous system, it's so slow that the vaccination can occur during that time and you can still live. But I think if you're bitten around the neck or chest area,
if you inhale an aerosol,
not going to do it. Yeah, the inhaler.
You have to. So within 7 to 21 days, typically depends on many factors, but you often have time. So she must have had them immunized, and the next day or two they got infected, which is surprising because you wouldn't think her dogs would contact others, but just walking. I'm sure she has someone walk them.
Right.
And they can interact with feces or other dogs.
It raises another general question of do you know of any other vaccines, especially viral vaccines, that can act after you catch the disease?
I do not.
Besides rabies.
I've actually been asked this before, and I could not find one other than rabies.
No, I think that's the only one. The vast majority, as you know, are childhood vaccines.
Yes.
And then there are a few for travelers and so forth, but none of them...
or bacterial. There's no bacterial vaccine that works that way either.
You know why this is? Because for most infections, you cannot identify the moment of infection. But with rabies, most of the time you can if you get bitten by a crazy bat or raccoon, right? That's a good question, though. I like that. I have to remember that for my virology course. Okay, that's it for parvoviruses. Next, Relenza-resistant H1N1 found in Australia. Now, this headline is incredibly misleading. You would read this and you would say, swine flu has been found to be resistant to Relenza. But in fact, it's not the case. There's a group in Australia who scanned a collection of older H1N1 samples that they had stored in the lab for resistance to Relenza, which is one of the 2 neuraminidase inhibitors. And they found a few that were resistant.
The lead giveth and the second paragraph taketh away. Yes.
That's right. Which article are you looking at?
I'm looking at the one, the resistant flu virus mutation found. It starts off, the H1N1 flu virus has mutated into a form resistant to the Australian-developed antiviral drug Relenza. And then in the second paragraph, we find out that this is a lab strain.
Right.
Sure. I get that all the time when I'm waiting online with my groceries.
Yeah.
You know, with the Inquirer. You open to the second page. Now, I haven't done that much, believe me. But if you do, you'll see that it's a totally bogus article and it's totally innocent on the inside so that they don't get sued for libel or slander.
We have, instead of the newspaper article, the original Journal of Virology article where this was published. And we know that in a journal, we don't do any such things.
Of course not.
We don't mislead people.
Never.
We put it all up front.
Absolutely. Especially in the materials and methods.
So Relenza is one of the 2 neuraminidase inhibitors against which there's been really no resistance in humans, or very little, if I remember correctly. You can make resistant viruses in the laboratory by passing viruses in the presence of the drug. But clinically, there's very little resistance, if any, to Relenza. Now, the other inhibitor, Tamiflu or oseltamivir, there's massive resistance, as you know. And it probably has to do with how these 2 work. These both bind in the active pocket of the neuraminidase, right, which is one of the 2 glycoproteins on the virus. And the neuraminidase is an enzyme. So normally sialic acid is the substrate that fits into the enzyme, and these inhibitors mimic sialic acid. But Relenza mimics it almost perfectly. It looks almost perfectly like sialic acid, whereas oseltamivir is slightly different, so you can get changes in the neuraminidase to make resistance very readily. So, right, the idea is that you can't change to be resistant to Relenza very readily because then you would not be able to bind sialic acid, which is an essential function for the virus. But in this study, they showed that a certain percentage of isolates, 2%, had resistance to Relenza, and there's a specific amino acid change that they identify. So it shows that it's possible. And these viruses grow well, and they're infectious in ferrets, and they transmit well in ferrets, so there doesn't seem to be any penalty with being resistant to the drug, which is interesting because apparently there's some way to change the neuraminidase in a way so that it still can effectively cleave sialic acid. And the makers of the drug, I noticed, were very quick to say that this has no clinical relevance because it's a lab study, right?
Yeah, right.
But in theory, it could arise. Right, I think if you can isolate such viruses in the lab, you might get them at some point. Now, remember, again, this is not the 2009 swine origin virus. This is older seasonal H1N1s. It could be that in the current strain it doesn't work, but we'll see. Nevertheless, interesting. Next, new HIV derived from gorillas. Alan.
Yeah. So this came out in Nature Medicine. New paper from Jean-Christophe Plantier et al. And this was a neat and kind of surprising to me, at least, paper, brief paper. So this group, they have, it's a French group, and of course they have one of those horrible government-run healthcare systems, so everybody's just in terrible shape. And as part of this system, they have this very rigorous surveillance for new strains of HIV. in this project, they just routinely have been checking anybody who has anomalous results in their HIV blood work. They go ahead and work it up and figure out what's going on, rather than just dismissing it as a fluke. So by doing this, by having this kind of surveillance going on, this network of French reference laboratories is set up to do this sort of thing. And at one point, a 62-year-old patient who had recently moved to Paris from Cameroon came in, and she was HIV positive. And they, in working up the strain that she had, they found they could quantify it with antibody assays and with some PCR assays, but then they had some other PCR assays that didn't work. They couldn't quantify the viral load with a group M specific commercial assay, for example. And so they went ahead and sequenced the thing, and got a nearly a complete genome sequence. And then they went ahead and constructed an evolutionary tree of it, and they found, based on this, that this seems to be a new introduction that is most closely related to an SIV strain that's found in gorillas. So it looks like a new introduction of HIV. This strain has not been seen in any other humans, and it appears to have jumped relatively recently from gorillas into humans. Now, this particular patient lived in an urban environment in Cameroon. She didn't seem to have any direct contact with gorillas. They don't mention whether she had a history of eating what's referred to as bushmeat in Africa. But it seems likely that she probably caught it from somebody else who had perhaps butchered a gorilla and picked the virus up that way. And, one of the things that I was wondering at that point was how many times has this virus jumped into humans that we don't know about?
Right.
Apparently she's healthy. She has no signs of AIDS, according to this article.
Right.
It has CD4 count of 300 cells per cc. Is that lowish? Does anyone know?
I think if it goes down below that, you start developing
below
symptoms.
300,
yeah.
Then you have symptoms. So it could be that this virus is not yet pathogenic in people. I don't know. It remains
What
to be
about
seen.
in gorillas?
Anyone know?
I believe that it's not in gorillas.
Not pathogenic, yeah.
It's like the SIV in chimpanzees.
Which, of course, recently has been now shown to be pathogenic in some chimps. Is it Beatrice Mintz? No.
Why don't I know that?
No. That's the transgenic person. What's the name? Beatrice Hahn. Sorry, Beatrice.
That's right.
Who travels to Africa and samples chimpanzees.
Vince was just mincing his words.
She has found that there are some chimps now that become ill with SIV chimpanzee strains of HIV. So,
well, you know what the problem with that notion is, in general, is that it's hard to get an animal in nature to live long enough to see the full manifestation of a lot of this.
Yeah, sure.
Because there's a lot of things that can go wrong in the meantime, like chimp malaria, for instance, and violent deaths and that sort of thing.
Well, the chimps, as you know, in Africa are on preserves, and they're cared for, they're left alone, and we know how many there are, and they can be studied to a certain extent anyway, and that's what she does.
Right,
she goes there and in very
These are
prescribed area.
rehabilitated chimpanzees.
I'm not sure if they're rehabilitated, but they're in protected areas, and she can go. They're all tagged, I believe, and you can study them. So she has done that for many, many years. And originally the idea was, yes, they didn't get sick, but just at this past ASV meeting in July, she reported that indeed some of them may become ill. So who knows if it's true for the gorillas. But is this something that we need to worry about, this virus, or is it just another example of how the virus has jumped from a primate species into people?
Well, I think it's a case of, if something happens once in nature, you can be sure that it's either going to happen again or it's happened before. It's just one of those things.
More support for the notion that it originally came from primates, closely related to us, I suppose. But whether or not it spreads in people remains to be seen, right?
Right.
And it's not clear how she obtained it, but there must be somewhere, at some point, the link that we don't know about.
So it raises the interesting possibility of if there are benign strains of HIV out there that can jump into humans, and you catch that one first, then does that offer some protection against the more virulent strains that you can catch from each other?
Man, it's a good question.
Just a
you know anything about that,
non-virology question here.
I really don't actually.
I mean, most people die, but there are survivors, right?
There are people who are long-term,
Right.
like long-term HIV,
Yeah.
never,
Right.
long-term no progressors.
Right, non-progressors. Right.
That's right.
So, I mean, can you tell if they're
Maybe they have.
they're infected a second time or not?
I don't know.
You know, there might be studies of that where you could see, a related virus that's different enough that you could pick up in lab tests, right? But I'm not aware of that. Could look that up, but offhand, I don't know.
Well, it sounds like they didn't know what they were looking for before.
Right.
In this case, yeah.
Right.
Yeah, sure. What do you think of that, Jen? Is this interesting?
I think it's interesting. I don't want someone to read it and think we're going to have a new outbreak of HIV spreading across the world.
Right.
But it does tell us that HIV is still evolving and it's an ongoing process.
Yeah, it's not just one introduction. Obviously, there are others. But did they make an estimate of when this happened? I guess this would be only the first time it's detected, but
Right. There's a they don't place any bets on that as far as I can see.
I would think from the sequence you could clock back and figure out when the jump occurred, right? I don't remember them doing that.
Yeah, I think they probably just steered clear of that.
Well, probably the bioinformaticians will take the data and massage it.
Yeah.
And I'm sure we'll have other isolates now that we know that this happens. Okay, last story is that the CDC, the Centers for Disease Control, wants the public to comment on this fall's vaccination campaign against the new H1N1 strain. Have you seen this? Anyone? There's actually a website you can go to and make an online reservation to attend one of several meetings in Colorado, Nebraska, Alabama, California, Indiana, Texas, Pennsylvania, Massachusetts, Allen, Washington, and New York. You can make a reservation to go. They want your opinion on whether they should do massive campaigns or normal seasonal campaigns. Now, I think that's very weird that they would ask a public that 23% of which cannot name a scientist what their opinion on is about the vaccination campaign. So I posted a blog about this on Virology, and I got fabulous responses from our readership, ranging from, I think it's a good idea, to, what, are they out of their minds? So I just thought we would chat about it, because it seems to me that they need to have a plan and just do it. And perhaps this is just a way of informing the public or making the public feel that they're involved, so that they're not saying, why do we have to take this vaccine, and not know anything about it. What do you guys think about this?
Well, it might be a good forum for the Centers for Disease Control to open up a general discussion about the value of vaccination and why people should get vaccinated for things that you can prevent by vaccination. It might ease a lot of rumors that are spreading that, mercuric thymol and stuff like this is causing autism from vaccinations in small children, for instance. There's a lot of fear out there now that's been spread, and we've had a show on that already, maybe several. So maybe the CDC is reacting to this backlash of public opinion about the value of vaccines to, okay, you want to talk about vaccines? We're going to have a big attempt to vaccinate for H1N1 in the fall. Come and open up the discussion, and let's see where it leads, because we can show you the value of what we're trying to do. Perhaps that's their motivation.
Yeah, I think it's a very good, probably good public relations move. Rather than handing down some edict from on high and saying these groups of people should get vaccinated immediately, they're going around and really, what I get the impression of is that they're selling the program and kind of getting people interested in what this vaccination would entail. But there's also the legal framework for this is if they decide that they want to promulgate some kind of specific regulations, for example, maybe requiring that healthcare workers get vaccinated. In that case, they would be required to put that to the public and put it out for comment as well. So they may be laying the groundwork for that, or at least keeping that option open by soliciting public comment this way.
I hope they're not going to base policy on the popular response to the question.
No. Usually, there's a very formal, ritualized way that federal agencies regulate. They go through this public comment period and all this, and they get a lot of whack nuts sending stuff in, but then they have to, they do actually have to respond to that.
Yeah, but they've never done it before for any of the other vaccines for flu. This is the point.
Right.
Well, this is the big first massive one, right?
And the flu vaccine is not mandatory.
No.
And they might be anticipating this big need to vaccinate a large number of people, and they need people on board with that idea.
That's
Absolutely.
It sounds like a good educational tool.
Yeah, I think it is too.
I think they shouldn't call it a, how the public feels about it. They should call it an informational session of some sort.
I think less people are going to go if it's an informational session than if they think their opinion matters.
That's true.
Yeah.
That's a good point, yeah. I think there are going to be lots of issues with this campaign, and it's probably a good idea, and I think that's a really good point. The way they named it probably gets people to come, although I would have gone anyway. I was reading a blog earlier today where this doctor said, the CDC wants all children immunized. That doesn't make any sense to me. They're going to die. Well, of course, it makes perfect sense, as Alan, and you remember the story we did a couple of weeks ago. This is the perfect group to target for immunization. So there's lots of misunderstanding.
That's who does most of the spreading.
That's right.
Doctor said that? That's incredible.
Well, I don't know if it was really a doctor,
Right.
This is a blogger.
There's a couple bloggers that are doctors that have some credentials for being a doctor, like New Age therapy sort of doctors that are spreading that vaccination is bad.
Well, also at the risk of causing domestic discord, although my wife doesn't listen to this, but just because somebody has an MD does not mean they know what they're talking about.
Here.
At least with vaccines, yeah. Yeah, I mean, other things, sure. I mean, there's a surgeon this week who said that you shouldn't get the vaccine, you should take vitamin D, and I'm not sure this is something a surgeon would know about.
No.
My father was a surgeon. He was a great surgeon, but when it comes to vaccines, nope. And he didn't know anything about it. He got his information from me. Yeah. So I think, the Internet's great, but it makes problems because there are a lot of opinions out there that are just uninformed, so you have to be careful. Right. And that's why we have TWiV.
You betcha.
We try and give you the facts. And you're going to see we have some emails that revolve around this issue. And so let's do some emails.
And when there are no facts, we try to discuss the controversies.
Yes.
And say, why aren't there any facts on this? Which is what science is all about. Then go out and find those. Make a hypothesis, go out and test it, and then come back and now tell us what you found.
But we need to reach more people still.
Yeah.
Okay, email. Allison writes, I love the show. Not sure how to address the question. Dr. Racaniello, Dr. Despommier, Vincent, Dick. Listening to all of the podcasts makes me feel like I know everyone on a first-name basis. First name is fine, right?
Absolutely.
No problem. Have you found that more people are interested in working in your lab since you've been doing the podcast?
Just the opposite, actually. We're losing one right here. Come on.
Well, she was already here. Maybe because I feel like if I were an undergrad in biology, microbiology, I'd want to work in your lab for my graduate work.
Oh, nice.
That's great.
That's nice.
So this is a PhD candidate, Allison at Drexel. No, I haven't noticed an uptick, but we haven't been doing it for a year even yet. And so I think it will take some time. It could be, but I'm not sure if that's the audience.
I'm not so sure that the PhD students are listening to this. No. I'm not even sure that the small virology department we have here knows it exists yet.
Right.
Right here in our very own backyard?
Yeah,
that's correct. I mean, Allison is a PhD student at a university, and she's listening. And there are a few others as well.
And I'm Vince's student, so I've only listened to a handful of these outside of his office doc.
It's not penetrated enough, and people's time is limited, I understand. But
That's true.
I suspect that it will increase. And then she says, I would love to hear more about any research which bridges virology and engineering. Absolutely. So last time, or 2 weeks ago, we talked about this.
Batteries.
Viral batteries, of course.
Right.
And then we talked about the pen-sized detector for viruses.
Oh, sure.
Remember? The gold filaments. So this is becoming more and more commonplace. There's a convergence to detect and diagnose virus infections. You need to bring in engineers. So we'll do that as much as we can. Thanks for writing, though. Raphael wrote, good day, TWiV hosts.
Sounds like an Aussie.
Is actually in the Philippines. I came upon a recent New England Journal of Medicine study entitled Severe Respiratory Disease Concurrent with the Circulation of H1N1 Influenza, and 3 other similar articles. What got me interested are, and I think we talked about those previously. 1, they call the virus swine origin influenza A virus, or SOIV. It was the first time I heard of this, and I even don't think you call the swine flu virus SOIV.
No, we did.
No, we don't. This is done because everyone was calling it swine influenza, and it's not. It's a human strain. So someone started calling it swine origin, which I don't think you need to do. I think it's, 2009 H1N1, or if you want to use the correct nomenclature. But you will find that in the literature.
The other thing is that they've identified a primary case down in Mexico, and it was a little girl, a little baby girl, and never had contact with pigs. So.
But clearly the genes are swine origin, though.
Oh, yeah.
Right.
I mean,
so that's
sure.
where this name is coming from. So from time to time I will use it, because I don't want to call it swine flu anymore, but lots of the press still does.
Yeah.
And as you'll see, CNN and NBC, they all do it. Number 2, the study also mentioned Thomas Francis's doctrine of original antigenic sin. Although I found this concept in Wikipedia, I also would like to know your comments on it, if there are any. Does this really hold true for all types of infections, or are there any exceptions? I checked out your virology blog for any mention of this, but I got an empty search result. Thanks for all the information you share in your blog and podcast. I do also share what I learned from you to my patients and colleagues. Well, that's great.
Wow.
Thanks, Raphael. Original antigenic sin. What a great name.
A fabulous name.
Jen, do you know what that is?
I actually looked into this a little bit because I had no idea what it was.
It's a fairly Catholic idea, don't you think?
Yeah. Does that belong in science?
I meant with a small c, please.
So my understanding of it was that the first time you are exposed to, say, a virus, your body mounts a memory response to that virus, and the next time you get infected with the virus, a slightly different strain of it, you actually mount a large response to the first virus, and not the one you just were infected with.
Right.
So it's original sin, as in your original, the one that you were originally exposed to.
Got it.
So this is really mostly with flu, because it's one of the few viruses that changes significantly antigenically.
And that you're repeatedly exposed to.
Yes. So you can get an H3N2 infection. Then if you're infected with H1N1, most of your memory is against H3N2. And this is thought why we don't have concurrent circulation of pandemic strains typically, but it doesn't always explain it.
A more scientific or engineering way of expressing this would be to say that the immune system exhibits hysteresis.
Hysteresis. Oh, nice.
You like that word, Dick?
I do. I love that word. I'm going to incorporate that somehow.
And what is it? What's the definition?
I'm going to phone up Alan after this show.
I was wondering the same.
No, hysteresis means that the system has a sort of memory, so its current state depends on its past states.
Yeah, cool. Well, he asks whether it holds for all types of infections. Well, it's been shown for some others, but as we said, flu is the most typical because you have this huge antigenic variation and you have repeated infections, so it's most common with flu. But Thomas Francis, a real giant in virology, actually, in the history of virology, first figured this out. There are a couple of old papers out there in the public domain. I'll put a couple of links in the show notes to those.
There's a newer paper you should put up as well.
What's that one?
I don't have it.
Well, get it to me. We'll put it up. Chris writes, I love the podcast. I'm a doctor of pharmacy, and I find the information to be very interesting and informative. I would love to hear something about JC virus. Recently, I saw a case where this virus was suspected. Thank you.
Jakob Creutzfeldt?
No.
John Cunningham virus.
It's a polyomavirus. Polyomavirus.
Sorry.
We talked about JC and other polyomas in TWiV 41 and 26.
Just talked about something Catholic, too. I mean, come on, we're getting pretty close here to
So
a religious experience.
JC virus keeps coming
exactly.
back.
When I was a graduate student, we joked about it, yes.
Well, it has some humorous connotations.
But polyomavirus is, we talked about serosurveys, where these viruses, polyoma, JC, BK, and there are a few others, infect most of us apparently, but we're mostly
Right.
well until we become immunosuppressed, and then they can start replicating more and cause disease.
Weren't they most recently seen in AIDS patients?
In AIDS patients, yes, absolutely. And if you get a organ transplant and you're immunosuppressed with a drug, you can get these exacerbations. And Rich Condit talked a bit about that. So a couple of recent surveys have been done just to see how much of the population is infected, and quite a few is. And I'm sure I will bet that if we took blood from the four of us here, we would be 90% seropositive for at least some of these viruses. But anyway, it's a good suggestion. We can certainly do more of a talking about JC and other polyoma viruses. Thanks, Chris. Matt writes, greetings. I just wanted to send all of you some well-deserved kudos for your superb presentation of TWiV, the engaging and informative weekly podcast on the world of viruses, the kind that make you sick, of course. Your banter is fun, but not over the top. And while you don't pull any intellectual punches with
That's because we edit out a whole bunch of stuff before we go on the air.
Well, you don't pull any intellectual punches with your use of nomenclature. Your explanations are thorough enough so that laymen like me are able to come along for the ride. Two viruses I'd like to hear more about are viral hemorrhagic fever, that sounds like a really nasty one, and Rift Valley fever. Thanks again, and keep up the great work.
Well, we did one on dengue, but we could go back and do another one.
How about a whole show on hemorrhagic fever viruses? You know, someone posted a question in my blog the other day, which is very interesting. He said, why is it that only RNA viruses cause hemorrhagic fevers? That was Everett. I have no idea. It's a good point, though. The DNA viruses do not cause hemorrhagic fever. Not that we know of. That's always the caveat. It may be some that we don't know about, but I don't know.
How many of the hemorrhagic fevers are vector-borne, besides the dengue type 4?
Yeah, they're all right.
Are they?
Oh, vector. Oh, insect vectors?
Vector-borne, yeah.
Sorry. Oh, that's a good question. Well, the Rift Valley
I was
is...
wondering whether I'd asked a bad question.
What's the vector of Rift Valley? It's a tick or a mosquito? We just talked about it, Dick.
We did. I know we did. I believe it's mosquito.
Let's look it up. Since we have the internet at our Rift Valley...
Vector.
CDC has a lovely page. Transmission to humans.
Mosquitoes.
Mosquitoes.
That's what I thought.
Yeah.
And sandflies.
Also
And sandflies.
But also...
also ticks.
But also directly handling the cattle meat.
Oh, right. Direct contact. Sure.
Yeah. So, Dick, I don't know the answer offhand. We can
Okay.
figure it out. That's a good question. All right. The next email is from someone called the peripatetic apoplectic.
It's peripatetic is a traveling person, yes?
It's traveling.
And apoplectic is somebody who's totally at odds with the rest of the world.
Okay. I have a gentleman and hopefully lady guest.
Yes.
As we do this time. I have a theory that for a given subject, e.g. virology, those in the know have their heads wrapped around the frontiers of knowledge, searching for the next discoveries.
Well, is that where our heads are?
Well, quiet. He's not finished. He didn't tell us where the rest of our bodies are.
The rest of, let's see, while those who do not know much about the subject find it onerous to try and obtain even basic knowledge of said subject. This void is happily filled by charlatans who the unsuspecting public follow and believe.
That's true. That is true.
Case in point is the ongoing mass hysteria on H1N1. I present to you some clips I took from a podcast with a wide audience, 400,000 downloads a month, in the hope that you will at least comment on it on your show. Sincerely, our peripatetic apoplectic. Okay, so we'll play a couple of these. These are from a podcast called No Agenda.
But no knowledge.
Right.
It is no knowledge, I'm sorry. It is by John Dvorak, who is a computer writer for many years, and a fellow named Adam Curry. Does anyone know who Adam Curry is? Adam Curry was the first video jockey on MTV.
I was just going to say,
Are you kidding?
Oh, I should know this. I think you mentioned it to me already.
Really?
So he's wealthy, and he has a website, Mevio, which
And that's how he gets his audience, do you think?
he has many podcasts, and he's just controversial. I mean, they're both controversial, and that's why they have an audience.
Something like Ben Stein.
And they shouldn't be talking about
Things they don't know about.
things they don't know about. So let's play a couple of these now. Let
Sure.
me make sure Alan can hear these.
Vince, for those in the audience, this is a radio show, so you can't see what just went on. Vince has a control board with blue, red, and white buttons, millions of them. And he said, let me just make sure that Alan can hear. And he reached over and grabbed the exact right button.
Well, we'll see if it's
I am
right.
incredibly impressed with Vince.
Let's see if I can...
I heard something.
All right, you heard something. Let's see if it's too loud. Here we go.
Very loud.
Here's the thing you have to ask yourself. Every flu that comes about comes out of Asia because of the nature of their agricultural system, where they mix the cows and the pigs and the ducks and the chickens. And so these things kind of mutate amongst those animals. How did this one come out of Mexico?
Aha. Okay.
I mean, how does this thing somehow evolve out of Mexico in the middle of the summer in a hot area like Mexico? I mean, the whole thing is a little fishy.
So, did you hear that, Did you hear that, Alan?
I did hear that, but the whole thing doesn't seem fishy to me at all. It seems to make a lot of sense.
Yeah, I mean, first of all, not every influenza has come from China. 1918 did not.
I gathered that he's looking for a conspiracy theory.
Yeah, of course.
Yeah,
Clearly,
I'm not really sure
clearly what he's
conspiracy
angling at
is.
is that,
Someone started this on purpose.
We did it intentionally.
Why
And
Mexico?
why would we make it so weak if we did it intentionally?
Exactly. Well, they're just testing it out for the next one.
My comment, the first episode we did after this was, if this is an engineered virus, I'm not impressed.
Right.
Yeah.
Ooh, I got some comments about that I have to send to you. The other thing is, in the hot weather of Mexico, well, in fact, the sequence shows that it probably started circulating there in December or January, so not that hot.
Can be cold there.
And, the whole thing he talked about, the confluence of animals and people in China, well, in Mexico and other places in the world, we now have that as well, correct?
That's true.
Agriculture is no longer, on that scale, limited to one country, so it's not surprising. So Mr. Dvorak and Curry should listen to TWiV and learn about virology and not get people all upset. It really is a shame that you have such a big audience, and that's where they're getting their science from, right? Now, I don't think we don't need to have 400,000 downloads a month, but come here for your virology.
I'm not so sure a music disc jockey on MTV knows all that much about viruses.
Yeah. I'll tell you, here's an interesting question we could ask ourselves, is that if you had the ear of the American public for 5 minutes, every single person, what would be those 5 minutes? What would you tell them in 5 minutes?
Oh, gee, that's too hard, Dick.
You think?
It would be difficult to keep their attention for 5 minutes.
Okay, that's true, but I mean, wouldn't you want them to know what the difference between a virus and a bacteria and a protozoan and a helminth and an insect and a... You could do that in 5 minutes.
Dick, I'd tell them where to go to learn about viruses.
I would want them to know about more than just viruses, though. I mean,
that's good. I mean, you could do the parasites. I would do the virus. We would have somebody
5
else.
minutes, that's all you've got. But everyone listens, and it has to be authoritarian. It has to actually have the authenticity of science.
Well, I could talk for 5 minutes and impact them about viruses for sure.
Yeah.
It seems like a very good exercise, you know?
I know.
We could do that, and each of us could read it on TWiV in the future.
So a 5-minute blurb on...
Yeah. Could you do that about parasites?
Sure.
No,
Just parasitism.
You're
parasitism.
a pro at talking to people.
38 years. Come on. It's
Some people do things for 38 years and never get it right.
You're right, but they didn't kick me out. That's the point.
Let's do one more clip just to...
And remember that, young lady. Your job is not over. It just started.
All right, ready for the next one? Here we go.
So this Guillain-Barré, there were lots of lawsuits, and of course the makers of the vaccine had to shell out all this money. So now they have been indemnified from any lawsuits should this experimental adjuvant kill people.
Right, and the adjuvant we were looking for is MF59. You can look it up yourself. And MF59 is a whack job of an adjuvant. They think it's going to do great things. And by the way, when you start looking into adjuvants, you'll find there's a whole slew of these from these different pharma companies that they're experimenting with. And the thing that they do is that they use them in animal medications. In other words, when your dog gets a shot, it probably has an experimental adjuvant that they're testing on these animals in real time, because these things are out there, and because you keep reading about, it's only used for animals. It's only used for animals. Why is it used for animals? Because they're trying to get clinical data so they can sell these, because this could be worth a fortune. Because the idea is, if you can find one where you can use your 1/10 as much of the virus, or the attenuated virus, you can really stretch. It's stretching the budget. This is the hamburger helper theory. And so they've got this MF59, which they're going to use, and that's why this is called experimental. The vaccine itself is not experimental. They just make them the same old way. But when you start doing weird stuff like this, now it's quote unquote experimental, and then they have to give them they have to indemnify them, and they do that too. And so God knows what's going to happen. This is basically, here's the way I'm seeing it. These pharma companies have seen that the FDA is holding pat on, we're not going to approve anything that's an adjuvant, because you don't even know how these things work. And so they said, since 1930. So they say, okay, let's do it this way. We'll make it, call it experimental. That way we can use anything we want because it's experimental. And essentially use the entire world's public to do a blind test on various adjuvants. There may be more than one or two different ones used in different areas. You may have the normal aluminum hydroxide in California, for example, and then they may use the MF stuff from Novartis, MF59 in Nevada. And so then they see how many people drop dead. And, maybe that one's a good idea. Who knows? I mean, as far as I can tell, this is a giant way to bypass, to do an end run on the FDA's kind of stubbornness about using these experimental adjuvants.
Okay.
Is it just me, or does this guy sound like he's 3 beers into the evening?
Wow.
Well, that's his shtick. He makes controversy. If you listen to his tech podcasts, he does the same thing. So he obviously has most of his facts wrong, and maybe we'll point out a few of them. MF59 is actually approved for use in vaccines in Europe. Okay, so he probably searches Google for MF59, but if you go to PubMed, you find all the dozens of articles where this has been studied. This is an adjuvant that has some oils in it. I think squalene is one of the components, yes.
Turns you into a shark.
Yes.
And these
Shark liver oil.
These have been tested. There have been extensive clinical trials. Nothing funny is going on here in terms of testing this. You don't get results from putting adjuvants in animals to submit for a human vaccine. That just doesn't happen.
And I don't think the FDA is going to take those results and be like, oh, you were vaccinating dogs without the dogs knowing it, and it's crazy.
No, this is totally crazy. What else is wrong? Anybody else got something? Where do you start? I just want to have a few concrete for our audience, because we don't want to just say this is garbage. We just want to say why.
I think it obviates the acknowledgement of a proper scientific method approach to developing anything. It just doesn't tell the truth about how the stuff comes about.
And the issue of indemnifying the companies, this comes up a lot in vaccine development, and in fact, it's the reason we have in many diseases where we'd like to have vaccines, we have no vaccine. It has gotten very difficult to interest companies in developing vaccines because of the liability, because the market is a one-shot deal in most cases. You vaccinate somebody and they never buy another vaccine from you again. And that's they are probably the most successful pharmaceutical products ever. But as a result, they're not very profitable. And then on top of that, you have this liability problem. It shouldn't be news to anybody that we live in a somewhat litigious society. So if anybody gets vaccinated with, for example, the H1N1 vaccine and develops any kind of problem in the 6 months after they're vaccinated, you can bet that they'll sue. And the companies are prepared to defend against a certain amount of that, but without any kind of indemnification to ask them to take that kind of risk and develop these vaccines and foot the development costs. It's not commercially possible for them to survive that way.
Well, they're talking about an older event that happened back in, I guess it was the 70s when they were...
the Guillain-Barré. They're talking about the swine
exactly
flu.
Right. And there were lots of Guillain-Barrés afterwards, and
That actually, yeah, I mean that was a real event that seems to have stemmed from the vaccine, and we still don't know why.
Nope, we don't. But the government did take a bath in the insurance. I know that.
Yes,
and that's where everybody got gun shy afterwards.
right.
I also want to mention that there are many new adjuvants being developed. This is not old technology, as he implied. He said it's from the 1930s. Well, there are brand new ones that are very interesting, and we do understand their mechanisms for many of these. They stimulate innate responses, which shapes the adaptive response. We absolutely know this, but I wouldn't expect them to know that because it's difficult to understand these things. And finally, none of these are going to be tested in people. And
people are not dropping dead because of the use of an adjuvant.
Right.
That's number one. I mean, nobody ever died from getting an adjuvant, no matter what.
I mean, not every flu vaccine that comes out in the fall is going to have adjuvant in it. And it turns out that the yield problems that some of the companies were having apparently can be surmounted now, so maybe we won't have to use adjuvant. And in any case, they will be trialed before they go into people. WHO has just issued a statement saying that vaccine safety will not be compromised. And I believe them, and the science says that that's the case. So anyway, that's two of the clips that he sent, and perhaps we'll deal with the others in the future. So let's move on to other emails. Swiss Compass, who sent a screen capture from CNN, which is them reporting on the, I think it was a Nature paper that we discussed a couple of weeks ago from Kawaoka and colleagues, showing that the 2009 H1N1 can replicate in the lower respiratory tract, cause a little more disease. And we discussed that. We said we didn't know what it means because in humans this virus is pretty benign. So the CNN capture, swine flu can ravage the lungs, spreads through respiratory system, causes lesions, doesn't stay in the head like seasonal flu.
Oh, wow.
Survivors of the 1918 flu are immune.
Seasonal flu's in the head.
Yeah, right.
Yeah.
You know, influenza is a lower respiratory tract disease, even seasonal flu. But this is in mice and ferrets and mice and miniature pigs.
Exactly.
Not in people.
Exactly.
It's just sensationalism.
Exactly. Slow news day. Sad that no man bit a dog.
Peter writes, gentlemen, I just listened to episode 41. It was great as always. Do you have any special plans for episode 50? That's our 1 year. Yes, we do.
I think we should do a spoof on This Week in Virology.
We
should invite these two guys,
they should,
and interview them.
Yeah, we have some plans. I'm trying to get a very special guest, but I don't know if it will work, if it will happen yet. So when we arrange it, we'll announce it. We do have some special plans. I wanted to send this link weeks ago. I forgot it. Meet the Scientist was the Science Podcast Pick of the Week in one of your early episodes. Meet the Scientist 30 with Dr. Stanley Plotkin might be of interest to other listeners. And Plotkin was a developer of the rubella vaccine, actually, and an old worker in virology. So we'll put a link to that show. That's quite interesting. Needless to say, Meet the Scientist can be subscribed to at iTunes, surely one of my top 10 podcasts, which of course includes TWiV. Best regards from Korea, Peter. Russell writes, I wish the media would stop with their unstoppable banter. MSN headline says, vaccine go-ahead given for unstoppable flu. Healthcare workers should be first in line in all nations, WHO official says. Well, it's more selling newspapers and getting web traffic. Everything is stoppable, right, Dick?
I believe so.
Even flu.
That's true.
But if you say it's unstoppable, people will listen.
They'll stop and listen.
Jim writes, check out this podcast on iTunes. Really love the podcast. Thought Dick in particular would get a kick out of this in a moment of entertainment. So there's a very interesting application for the iPhone called, what is it called? Darn, now I forgot the name.
That's a good one.
Anyway, basically you can record short podcasts on your iPhone. They automatically get uploaded to a website, and then you can go there and listen to them. It's a free app, and it makes you a podcaster, although you can only do 2 to 3 minute podcasts. Anyway, someone uploaded a rant about if at his workplace, if you sneeze 3 times, they make you go home and they penalize you for having
three is a magic number for me.
I'm only a 2.
You're a 2 person?
Yeah, I never get to home.
My wife always sneezes 3 times. I wait before I say Gesundheit.
Boo, audio boo. Thank you. Audio boo. Turned anyone to a podcast. So why did he think you would like this in particular, Dick? Anyway, thanks, Jim.
Don't know.
And we have two more.
Might think I like to talk.
Arius
Yeah, I'm
writes.
guessing so.
He sent us a link to an AP news story. Apologies if you received 100 copies of this already. Government is calling for several thousand volunteers to roll up their sleeves for the first swine flu shots in a race to test whether a new vaccine really will protect against the virus before it is expected to rebound in the fall. So anyone wants to volunteer, you can volunteer for these studies. They will begin in 8 medical centers around the country: Baltimore, Iowa City, St. Louis, Nashville, Seattle, Atlanta, Cincinnati, and Houston. Not New York.
No.
Or Western Massachusetts.
No.
Back when I was in college in Iowa, they asked for volunteers for the smallpox vaccination.
Really?
Yeah.
Did you go?
I couldn't because if you were around anyone that had eczema, you couldn't be one. And boyfriend at the time kept me out of $200.
Darn.
Well, I received that.
Ex-boyfriend.
Yes. Ex-boyfriend.
I got that vaccine as a child, that smallpox. All right, the last email is from Stephen. Keep up the great podcasts. I really enjoy them now that I have time to fall down rabbit holes to listen, read, and explore. Even I don't know how I ended up here. And he sends a link for a T4 bacteriophage plush toy, which brings me to the picks of the week. And I'm going to go first because my pick is giant microbes. Thanks, by the way, Stephen. It reminded me of this website where you can buy plush toys of your favorite pathogen. You know about these guys?
Giant microbes. Is that like jumbo?
Yeah.
They come in different sizes too.
Yeah, we all have some. I have a polio. You have some in the lab.
I have a bunch. People give them to me as gifts.
Yes, the people who work on these,
yeah.
people figure it's a good gift. My students always get them from their mothers. Do you have one? You know about this, Alan?
No, but I'm looking at the site. It looks great.
Yeah, it's fun. They have viruses, bacteria, parasites. They made a swine flu virus, which is
MRSA is adorable.
They have, it looks like a pig. Which one is adorable?
MRSA,
yeah. Anyway,
yeah, I like
giant
that.
microbes. It's pretty. It's a lot of fun. It's a little.
I meant to say
levity.
jumbo shrimp. That's probably what I meant to say.
So let's continue with our picks of the week. Jennifer, what have you got for me?
I wanted to pick the PhD comics. So my fellow grad students and I read them all the time. They're by George Cham, I believe, who got his PhD in medical engineering at Stanford. And the comic strip is called Piled Higher and Deeper. And it really just lets us graduate students laugh at ourselves. There's some real truisms where you don't feel quite so alone in the graduate school life.
Hear, hear.
Yeah, that's a great one. I love that. I think you've seen that too, Alan, of course.
Yes, absolutely.
So that's Jennifer's parting donation from the Racaniello lab.
But you never got to where she's going.
We'll get to that.
Oh, I'm sorry.
We'll get to that.
Sorry.
Dick, what have you got?
Well, it's an ASM publication called Microbe, and in it has the complete history of virology. It's called Virology for the 21st Century.
Very good.
And it's sort of a blocking on this word here. Why would that ever happen at this particular moment? But it's an executive summary of an executive summary.
Very
That's
good.
what I meant
Dick,
to say.
You're never at a loss for words.
Well, I was just at a loss for words. It just happened, but I suddenly recovered.
Very good.
So that's I read it, and it was somewhat like the one we read on the air, but there were a few interesting omissions.
Virology in the 21st Century. Very good. Thanks for that. And Alan.
Well, my pick of the week is a book that I've been reading. It's called Annals of the Former World. It's by John McPhee. This has nothing to do with virology, but it's about geology, which
is great.
As a biology, chemistry major in college, and then going into microbiology, I completely missed geology. And it's an absolutely fascinating subject. And John McPhee is just one of the best science writers ever.
Right.
So he does a brilliant job of explaining this stuff. This is actually the book or the series of books, it's now bound under one cover, that made him famous, that won him a Pulitzer Prize. It's an astonishing piece of work. It took him about 20 years to write, over a period of several volumes. But don't be intimidated by the thickness. It's actually, once you get into it, it's a gripping read. It's just really, really neat stuff.
Very good. I'll have to get that one. I have a long list of books now that I have queued up. Any of you read books electronically, like on an e-reader?
Not yet.
I hear next year is the year.
Everyone
I've
will
been
have
hearing
a Kindle.
that for several years.
We'll have a Kindle. I know they're very concerned about this at the New York Times, for instance.
Yeah, we talked about that with Delthea.
That's right. Well, that was Newsweek. But this is the New York Times is really, they're concerned that in another 2 or 3 years there will not be a newspaper out there.
Well, they can publish on the Kindle.
Well, they will, of course.
That's fine. Okay, that's it for our picks of the week. And that's it for another TWiV. Jen, thanks so much for joining us. Really appreciate it. So where are you going? Now, next week is your last week.
It is.
In the lab. What are you doing next?
I'm going down to Washington, D.C., and I'm going to be working for a company doing research into the best case practices for hospitals. I'm currently thinking about the last big experiment I'm doing in lab,
Okay.
But I'm wanting to, move away from the bench for now and start really thinking about public health issues and how we can, for lack of a better word, use like sort of the translational research, using some of the things that we know about viruses and how does that play out in the real world in hospitals and how can we sort of protect the public and fight infections and things of that nature.
Do you think you'll miss the bench?
I didn't think I was going to until like the last month or so as I'm winding things down and I'm thinking, this is the last time I'm going to smell E. coli in the morning. This is the last time.
I love the smell of E. coli in the morning.
Right. I just thought that
not going there. I'm not going there.
So we'll see. I mean, there's a chance I'll make it back to the bench.
That's great.
And I don't think I'm done learning yet.
Of course.
Certainly not.
Suggestion from somebody who left the bench a number of years ago: take up cooking.
I'm an avid cooker. So
Okay.
Alan, I'm going to send you my my cookbook. It's called Scientist as Chef.
Ah.
It's shovel ready.
Well, Jen, you may not miss virology, but we will miss you. So perhaps
I'm going to miss virology. But I'll start really listening to your podcasts and reading your blog.
Well, once you have some experience there and you know this different world, we'll have you back and you can tell us
Exactly.
what you're doing. That'll be interesting.
Yep.
Sounds good.
Confessions of an ex-virologist.
That's right. Alan, thanks to you as well.
Always a pleasure.
Of course, always good to have you. And we'll look forward to next week. Alan can be found at dovedocs.com. You can also follow him on Twitter, Alan Dove, all one word. Dick, thank you.
Pleasure.
Dick is at tricanella.org.
Yes.
Thanks for coming. Always good to see you as well. And you can follow TWiV on Twitter or FriendFeed at PROFVRR. We have a website, twiv.tv, where you can find links to all the articles that we talk about, show notes, and other interesting tidbits like the MP3 clips that we played today. So do check that out. And, of course, do send us your comments and questions and desires. You can send us email at twiv@twiv.tv. You can send us an audio file, and I just received the second this past week. We'll play in the future. Or you can leave us a message on Skype. If you call up Skype to the account TWiV Podcast, you have about 10 minutes to leave a voicemail. Tell us what you want us to talk about. Ask us questions and tell us what Dick got wrong. Actually, all of us, we all get things wrong. I like to pick on you because you always laughed.
Vince, I came in knowing that I had it wrong, so it doesn't bother me at all.
No, you didn't have it wrong. You have been listening to TWiV, the podcast all about viruses. And you can find us not only at twiv.tv, but also on iTunes. And why don't you go there and subscribe? It's free, even though subscribe sounds like you're paying money. It's not. You just click a button. You can search for TWiV. You'll find it. Click subscribe. As far as I know, it's the only podcast that's all about viruses, the kind that may or may not make you sick. Thanks, everyone, for listening. See you next week. Another TWiV is viral.